with a high mortality rate above 85% nowadays. Now, the only effective therapy of fulminant hepatitis is urgent transplantation of liver. That is the reason why today a lot of experimental models are used in order to find a new and effective therapeutic agent that will replace transplantation as the only effective therapy nowadays. I will present here Cone-induced liver injury. That is a well-established murine model of autoimmune T-cell mediated hepatitis, and this model resembles the pathology of autoimmune hepatitis in humans. Maybe some of you will find this interest to make any mathematical model of this disease. Okay, first of all, what is Cone? Cone is an abbreviation of concanaval A, a molecule that is extracted from the bean, and it is for more than 50 years used in immunology as polyclonal activator of T lymphocytes. But during last 20 years, this model represents an excellent model of acute liver failure in humans. When you inject Cone in mice, when you inject Cone in mice in different doses, from 10 to 40 milligrams per kilo, 40 is the lateral dose, to use 12 milligrams, Cone goes to the liver. And what happens in liver? Cone was captured by liver-resident macrophages, Kupffer cells. They take Cone, then activate, and then produce a large amount of pro-inflammatory cytokines, TNF-alpha, interleukin-1 beta, interleukin-12, and interleukin-6. So, activated macrophages produce pro-inflammatory cytokines, and as hematoxins, a lot of cells from the circulation migrate in the liver. At the same time point, Cone, as I mentioned before, is polyclonal activator of lymphocytes, particularly CD4 positive helper T cells, cytotoxic lymphocytes, as well as NKT and NK cells. So, next step during pathogenesis of this disease is that pro-inflammatory cells migrate in the liver, and then in the liver they destroy hepatocytes. They destroy hepatocytes directly by cell-to-cell contact or by producing pro-inflammatory and hepatotoxic cytokines, among all the best defined role of TNF-alpha and interferon gamma. So, result is mass infiltration of pro-inflammatory cells in the liver and apoptosis or necrosis of hepatocytes. And all this happened very quickly during 2 to 8 hours in the liver of mice. So mice after 24 hours has severe acute liver failure. Since all these cells, NKT cells, macrophages, CD4 cells, hepatocytes by themselves, express two molecules important in immune response, two molecules which are ST2 molecule and galectin-3, we decided to use this experimental model to study the role of these two molecules, ST2 molecule and galectin-3, in the induction of inflammatory pathology of the liver. So first I will present our results regarding interleukin-33 ST2 axis in this disease. Only short introduction regarding the ST2 molecule. ST2 molecule is present in two forms, soluble form and membrane-attached form. Soluble form is decoy receptor for interleukin-33, and there is only few papers describing its role. Its role is that it binds to membrane receptors of macrophages, down-regulates toll-like receptor 4 expression, and attenuates production of pro-inflammatory cytokines, and by that it reduces inflammation. On the other hand, ST2 membrane form is attached in the membrane of a lot of immune cells. It is attached by its receptor accessory protein, interleukin-1 receptor accessory protein, and interleukin-33 binds to this complex ST2 interleukin-1 RAP. This complex, or ST2 molecule, is expressed on all immune cells and all effector cells that are important in this disease. What happens when interleukin-33 binds to its ligand ST2 molecule? Well, the dominant effect is production of large amount of Th2 cytokines, interleukin-4, 5, and 13, and downregulation of Th1 and Th17 immune response, or downregulation of pro-inflammatory cytokines TNF-alpha and interferon gamma. The other important mechanism of interleukin-33 is its action after necrosis, after cell necrosis. So, if cell die by apoptosis, interleukin-33 remained captured in apoptotic bodies and was demolished by, degraded by caspases. If, on the other hand, if cell died by necrosis, from necrotic cell, interleukin-33 will be released and will be, then it acts its way, it binds to ST2 ligand and promotes healing or promotes Th2 immune response. As interleukin-33 ST2 signaling pathway is involved in the suppression of Th1 cell-mediated immune response, and I mentioned before that Th1 immune response is important in chronic hepatitis. ST2 is expressed on the pivotal or major effector cells that mediate liver injuries, such as T lymphocytes, NKT cells, macrophages. We aim to investigate if interleukin-33 ST2 axis has any role in pathogenesis of Coney-induced liver damage. Now I will present our results. First of all, we use histology and biochemical analysis between wild-type mice and ST2 knockout mice. Wild-type mice were on bulb C background, which is relatively resistant to Coney hepatitis. As you can see here, ALSTA and ALTA levels were significantly low compared in wild-type animals compared with ST2 knockout animals. Next, histological analysis showed that healthy Coney untreated mice, there is no difference. But when you compare histological samples of ST2 knockout mice only 24 hours after Coney with wild-type mice, you can see that there is huge number of necrotic fields with total percentage which is 5 or 6 times more than wild-type animals. Bulb C background, relatively resistant to Coney. So the first conclusion will be ST2 knockout mice are more sensitive to Coney-induced hepatitis. Now we investigated why. First, we checked pro-inflammatory cytokines in sera, and we found out that serum levels of all pro-inflammatory cytokines, among them TNF-alpha and interferon gamma is most important. These cytokines were significantly higher in sera of ST2 knockout mice. These are control animals, 0 hours and 8 hours animals, which showed that TNF-alpha and interferon gamma were significantly higher in ST2 knockout mice. And what is very important, interleukin-4 was lower ST2 knockout mice. I mentioned that interleukin-4 as representative of Th2 cytokines is important for this axis. Next, we focused on target tissue, liver, and we want to find out what happened in the liver, which cells migrated in the liver. We find out that ST2 deletion upregulates total number influx of all important effector cells. So, total number of mononuclear cells, T lymphocytes, cytotoxic T lymphocyte, helper T lymphocytes, B cells, macrophages, NKT cells, they were all present in higher number in ST2 knockout mice. Only T regulatory cells, CD4 positive, CD25 positive, FOXP3 positive cells were less present in ST2 knockout mice. These cells are important because these are immunoregulatory cells that have protective role in this disease. Okay, cells, influx of these cells were higher in these animals, but then we want to know if intracellular staining of these cells would relieve which cytokines they produce. And we find out that the analysis of CD4 positive population showed that there was significantly higher number of CD4 positive TNF-alpha producing cells and less number of CD4 positive interleukin-4 positive cells. So ST2 knockout mice has disbalance in Th1 on Th2 immune response towards Th1 response as dominant one. Then we find out that also interferon gamma producing CD8 positive cytotoxic lymphocytes, NK cells, NKT cells, as well as interleukin 17 producing NKT cells was significantly higher in the liver of ST2 knockout mice. So, the first conclusion was ST2 knockout mice were sensitive to chronic hepatitis because there was a significant disbalance between Th1 and Th17 immune response, which were responsible for hepatotoxicity in these animals. But then we decided to investigate if interleukin-33, recombinant interleukin-33, will have protective role. For that purpose, we use recombinant interleukin-33, and we gave it 24 hours or 12 hours prior to Kone injection. And our results show that if you use recombinant interleukin-33, you can protect mice liver from the damage. 1 microgram of interleukin-33 in one dose given all 12 hours or 24 hours before Kone significantly attenuated disease. Levels of IST and ALT was significantly lower in these animals compared to Kone-treated animals. Histological samples show that livers of these animals are very similar to healthy controls. As we did before, we also measured inflammation. By measuring pro-inflammatory cytokines, we showed that interleukin-33 managed to significantly reduce TNF-alpha, interferon gamma, and interleukin-17. On the other hand, the level of interleukin-4 was significantly higher in the sera of interleukin-33 treated Kone-treated mice. So interleukin-33 ST2-axis managed to disbalance immune response towards Th2. Then interleukin-33 favored Th2 response in the liver and significantly reduced influx of all mononuclear cells, but particularly those that are aggressive, CD3 positive T cells, cytotoxic lymphocytes, CD4 positive T lymphocytes, and augmented significantly protective CD4 FOXP3 positive immunoregulatory T cells. Then we also, by intracellular staining, revealed which cytokines produce these cells. We showed that these cells produce TNF-alpha, interferon gamma, and significantly more produce interleukin-4. Of course, TNF-alpha and interferon gamma were less than Kone-treated animals. Next, also total number of interferon gamma producing and interleukin-17 producing CD8 positive T cells and K-linked T cells was significantly lower in interleukin-33 treated mice. So first conclusion was interleukin-33 ST2 signaling pathway moved the immune response towards Th2 and has protective role. As I mentioned in introduction, interleukin-33, when it is released from necrotic cell, it will act as an alarm. So we would like to see what happens with apoptosis in the liver of these animals. Interleukin-33 suppress the expression of pro-apoptotic and induce the expression of anti-apoptotic genes in Kone hepatitis. As you can see here, percentage of cells that express active caspase 3 was significantly lower in interleukin-33 treated group. And in interleukin-33 groups, the activation of pro-apoptotic bugs and enhance the expression of BCL2 and PERK. So interleukin-33 suppress pro-apoptotic and enhance anti-apoptotic genes in the liver. So our first conclusion was interleukin-33 ST2-axis has protective role in experimental model of fulminant hepatitis. These are mechanisms: prevention of Th1 and Th17 immune response, and prevention of apoptosis of hepatocytes. This work was published in Journal of Hepatology, and after that, the professor Giza-Tiggs, which invented this model of chronic hepatitis, wrote an editorial on this paper, and she proposed the mechanisms of this signaling pathway. So, Kone was injected, macrophages, Kupffer cells produced TNF-alpha interferon gamma, Kone activated TNNKT cells migrate in the liver, produced cytokines, and cell-to-cell contact induced cell death. After cell death, interleukin-33 was released from liver sinusoidal cells at hepatocytes, captured to ST2 receptor, and Th2 immune response leads to healing and regeneration of the liver. Second part of this lecture was the role of other molecule, galectin-3, in the same disease. I will try to be as quickly as possible. Galectin-3 is a member of galectin family. Galectin family has several molecules. Galectin-3 is important for migration of mononuclear cells, for chemotaxis, for apoptosis. If it is membrane bound, it will induce apoptosis. If it is present in the cytoplasm, it will inhibit apoptosis. It is also important for the function of all effector cells, including macrophages, dendritic cells, T lymphocytes, NKT cells. It is also involved in inflammatory diseases and malignant diseases in the liver. So, since it is expressed in all cells of the immune system involved in chronic hepatitis, and till this work was published, its role in pathogenesis of pulmonary hepatitis was unknown. We investigated its role in this disease. Before we started this project, we tried to find out if there is any connection between expression of galectin-3 and human liver pathology, human liver hepatitis. As you can see here, normal liver, there is very small or no expression of galectin-3, but this is the tissue sample of liver of patient suffering from isoniazid-induced hepatitis, and this is the liver of patient suffering from hepatitis B. As you can see, galectin-3 was highly expressed in the livers of patients suffering